Somatic cell nuclear transfer is the method of cloning in which the nucleus of a somatic cell is placed in an enucleated egg cell. The success of this method of cloning proves that old cells retain all the information necessary to be young. It also proves that there is something in the cytoplasm of egg cells that can rejuvenate old cells. In 2006, Yamanaka discovered 4 proteins in the cytoplasm of egg cells that could induce fully differentiated cells into an embryonic-like state. Nelly Olova is a researcher at the University of Edinburgh who's going to tell us about partial reprogramming, which is a method of rejuvenating old cells with such germline factors. Reprogramming. So let's all welcome Nelly Olova.
Thank you, David, for the introduction and for inviting me to be here. It's a pleasure to be with you this weekend. I'm going to go over the things that you mentioned as well. And I made a bit more generalist introduction because we saw yesterday there are many mathematicians, chemists here, so it'll be good to know what is what. Hopefully it'll answer some of the questions about DNA methylation you had earlier. So I did my PhD on DNA methylation back in Cambridge with Professor Wolf Reich. So biologists maybe haven't heard about him. He spent years studying DNA methylation. He found out that that's the basis of imprinting and some out of the things that DNA methylation does, and investigated early embryonic development for many years, which was also the area of my research when I was in his lab. That's him, and he recently became director of the Altos Lab, Cambridge Institute, to work on rejuvenation and anti-aging therapies. And after 7 years in his lab, I moved on to the University of Edinburgh, where I'm a research fellow now in the group of Tamir Chandra. His work has been in senescence, the epigenetics of senescence and chromatin organization. We do a lot of single-cell transcriptomics, work a lot on epigenetic clocks, and more recently rejuvenation. So it did seem like a very long stretch for me in the beginning to come from early development and to go on to work on aging and senescence. It was, I couldn't even find a plot that would go from zygote to an old person because, I worked on zygote and embryonic stem cells. But actually there happens, now we know that there's more common and that these two fields kind of meet together, and it's very exciting with the partial reprogramming that I'm going to talk about. So I'm going to tell this from kind of the perspective of my journey from the epigenome and what DNA methylation is and what it does. And I've put this, starting with the genetic code for all the maths and chemist people. So we know we have those 4 bases that build the genetic code, A, C, G, and T. And the genetic code is more or less different for every person, but very similar in the humans overall. While the cells in our body have the same genetic code, but they have different epigenome. So the instructional manual for the activity of the DNA, the genes, is in the epigenome. So that's epigenome, it's above the genome, and it regulates the genome. And that includes the histone, packaging, and marks, and all the other protein remodeling the genome, including the DNA methylation marks, and also some like non-coding RNAs, which nowadays also very, very popular to study. And so what is the DNA methylation, what it does? I'm glad that there are chemists here because, it's a chemical modification on the DNA, and it's it's a site like a methyl base forming a C2C bond, which is very, very strong and not easy to break. It positions itself, unlike most schematics that make it like a lollipop sticking out of the DNA, it's actually inside the DNA, between the two strands. And because we already mentioned it earlier, it's usually found on CG diodes. So it's a CG dinucleotide that is faced on the other side of the DNA strand with another CG dinucleotide. So they form these CG diodes. And usually they're methylated on both symmetrically. So you get these two methyl groups in the middle that strengthen quite a lot the bond between the two strands, and they change a lot the physicochemical properties of the DNA. They make it much more rigid, hydrophobic, so less soluble, and then much more less accessible, and the two strands become much more difficult to separate in order to replicate or read. So that's all from chemistry papers that usually biologists don't have never seen, but you can see now people speak about chromatin phase separation, liquid, and others. So DNA methylation has a lot to do with that for sure. I've added this because we discussed also about the enzyme, and just kind of to give the full picture. So the vast majority of the, did I skip anything? Our genome is methylated. And there's a small handful of CPG sites which are not, usually in those called CG islands, which we also mentioned earlier. And DNA is deposited by enzymes that first do de novo, deposit the marks de novo, and then with each cell division, with another enzyme, they're easily propagated. Because of this symmetric dyad, it's easy from one strand to replicate the mark on the newly synthesized strand against it. So there's an enzyme that just copies the mark from a new strand, from the old strand during replication to the new strand. So basically there's a very accurate and good mechanism that can propagate this mark through self-divisions. So it's an overall a very stable mark, and with the properties that I already mentioned, that kind of helps it to do the functions that we think it's doing. It's known that it's a repressive mark. So while this plot is conditional, it's a bit old plot that the CG island, when it's not methylated, it's expressed. When it's methylated, it's not. And while the lower is true, and now we know that that's more valid for enhancers rather than promoters, when you have unmethylated promoters, there are other mechanisms to suppress gene expression, so that's not true. But if you only think of DNA methylation, that's what it generally does regarding gene expression, and it definitely does this for the repeats. So a large proportion of the mammalian genomes are made up, in general of eukaryotic genomes, are made up of repeated sequences, and a lot of them, if not mobile, are transcribed. And this transcription is most of the time not good for the stability of the genome. So what DNA methylation does is that it packs all the repetitive elements and keeps them silenced, shut down. And then this leads to the global role that it has in the body, which is to set the cellular programs. So the same DNA that each cell has in our body, in order to do the different functions that the different cells do, which are part of different organs, then they have to have different sets of genes turned on, and that's what DNA methylation, together with the other chromatin, elements of the chromatin, do. But a lot of it is dictated by the DNA methylation. And there are more than 200 cell types in the human body, and as a rule of thumb, they all have a stable epigenome. So once these marks are set early in development, they don't change. Like, they're not supposed to change. I'm just stressing on this because, especially with the epigenetic clocks, many people start working on DNA methylation. And I see in papers or in talks, they're like, oh, DNA methylation is a dynamic modification during lifetime. Well, it's not. It's supposed to be there and to stay the way it is. And in the instances when it changes, that leads to pathology or aging. And yeah, the pluripotent cells have plastic genome versus the differentiated already somatic cells that have stable epigenomes that don't change. And the setting of the somatic programs happens very early in development. As you mentioned, the zygotic stage up to the blastocyst, so that's before implantation. Even so, that's where we have a big wave of erasure of the parental methylome, because the cells come together with an identity of sperm and an egg. And you have to erase this identity to form a totipotent embryo that can become any cell type, and not just those two. So globally, the methylome is erased, and it in different ways on the paternal and maternal compartment of the DNA, which are here in red and blue. And then it erases over the first few divisions and begins to be set, like in the newly formed embryo, already just before implantation and around implantation. So that's basically in the first week for the mouse, and maybe in the first 10 days, couple of weeks for the human development. And you need, activity of the enzymes that both set the marks and remove the marks as well. And when this fails, you have early developmental failures or syndromes. And it's not actually easy to remove the mark. I mean, chemists would know that, yeah, you can't just remove the methyl group from the nucleotide. And there are ways around that. You cannot just directly remove, and for a long time they sought for such enzymes, but they don't exist. So what can happen is DNA, either the methylated cytosine can be oxidized, which is the mechanism that was discovered 2009, so not that long ago. And there are 3 stages, to oxidize it from hydroxy to then formyl or fully to carboxylated group. And then these can be removed by either stay as they are or be removed. But the hydroxylation already on its own changes all these physicochemical properties that the methylation causes. So even if it stays just as hydroxy, DNA is already more accessible, not as rigid, and can be read and open. So it reverts the physicochemical effect of methyl without actually removing the mark. But there are also ways to remove it, and that is to remove the whole nucleotide. You can't get rid of it otherwise. So that's with repair mechanisms. And they're mentioned here through TDG or AID. These are different repair machineries. So they either remove the oxidized mark or the nucleotide, with the nucleotide with the oxidized mark or with the methyl itself. But these mechanisms generally work only during replication. So they're limited to dividing cells, which is the case with the early embryo, but not with somatic cells, where most of them don't divide. So apart from oxidation, a lot of the demethylation machinery is not available in somatic cells. And of course, there's the so-called passive demethylation, which is the main way to do the big global remodeling in the beginning, just not to copy methylation on the new strand. And while you replicate with a few cycles of replication, if you don't copy the marks, then the marks dilute, and you've achieved quick demethylation on a very global level. So they're supposed to stay stable in life, just because a lot of the machinery is not available even in adult cells. And because only, the DNMTs, not all are active there. The enzymes that oxidize, one is active later in adult cells and not in all. So if that happens, if there are changes with the methylation programs that instruct the cells, so then the cells become cancerous. One of the things, they like go mad and they don't know what they're doing. They start self-propagating or other pathologies, and now we know aging as well. So you can see why like cellular differentiation is irreversible, and here is something that early developmentalists always show, the Conrad Waddington's epigenetic landscape, where he made a metaphor of the cell states being marbles that roll down valleys between hills. And these are the barriers that the cells have to overcome to become another cell type or to return to to pluripotent states. And as you see, these are the epigenetic barriers that are set by DNA methylation in the chromatin packaging. In order to change these, it's not easy, it's not trivial. And it happens only under certain conditions, very early in development. But of course, then in 2006, Takahashi and Yamanaka managed to find factors that can be brought into the cell and turn on this program, which has been shut down in the adult cells to be never turned on again, and which is only basically brings back this big global remodeling that happens only early in development in the living organism. So they introduced these 4 factors, and CIMIC out of them is also like an oncogene. So they help the cells to start dividing again, to remodel their epigenome and become pluripotent. So they become again with a plastic epigenome and lose a lot of the marks, like the programs that they have been set to have at this time. If it's a skin cell, which is what they took first, then this skin cell is not more a skin cell, is not anymore a skin cell, but a pluripotent cell. So basically then the Waddington's epigenetic landscape is revised. Nowadays we can actually go back and do the things which cannot happen spontaneously in the body, or if it happened would be a problem. And while everyone thought, Yamanaka and Takahashi did that to get pluripotent cells, and as David mentions as well, the first attempt was actually the cloning, and much earlier by John Gurdon with the frogs. And in 1996, the first mammal was cloned. So that's Dolly the sheep, which is now in display in the museum in Edinburgh. So everyone can see her if you wish and go to Edinburgh. So although Dolly the sheep died quite early, so for this breed, they live up to 12 years, and she died kind of halfway and had some health problems. But all the other cloned animals after Dolly lived normal lifespans. So one of the big questions then that was already proven with Dolly and the somatic cell nuclear transfer, which preceded the Yamanaka factors reprogramming, is that there was this ability in the oocytes to rejuvenate the cell. So if you have a cell from a 6-year, I think her mother cell was 6-year-old, then maybe with Dolly she didn't manage to rejuvenate, and that's why she died earlier. But they're able to revert all the aging marks so that you start from 0 and reset, and so that you can have another full lifespan coming from that cell. While if that cell remained with the body, that would not live so long. It was the mammary gland cell. So the idea that cloning does rejuvenation was already shown then, and in 2011 it was published that you can do this, you can rejuvenate with reprogramming but again going through the pluripotent state. So as Prim Singh mentioned yesterday, with the cloning you go to the embryonal stage, and then you're rejuvenated, and then there's another new organism that's rejuvenated starting from zero. And this study proved that you can do rejuvenation with the Yamanaka factor reprogramming, but again, passing through the pluripotent state, and then these cells, when they're redifferentiated, they actually have features of a younger cell. And Prim Singh came with this idea, as he said, he could draw on the back of an envelope at some point, because he was working with the team that created Dolly the sheep, that it would be possible to do rejuvenation without passing through the embryonic stage, but kind of just before that, maybe to introduce factors which are present in that ovum with the cloning that can rejuvenate without actually going through the embryonic stage. And he did his proof of concept a few years later, where he took human primary fibroblasts and introduced the 4 Yamanaka. So it was actually 6 factors, because he took senescent cells. And the previous study that I showed that 6 factors are necessary to reprogram senescent cells, which are in a different type of cell cycle arrest than the normal somatic differentiated cells. And he managed to show that one factor, chromatin factor HP1 beta, the levels got restored back to normal, to levels of a young, or like non-senescent in this case, fibroblast. So he only checked this, but it was a proof of concept, and that's part of the chromatin as well. So for him, he believed that's epigenetic rejuvenation and is based on epigenetic factors. When these 4 factors are expressed in vivo in an organism, though, it's shown that if you express them and leave them, they quickly form teratomas because they have cancerous properties, the embryonic cells. So it was dangerous to express these factors in a in vivo organism. But what was published in 2016 by Belmonte's group was that they expressed transiently these 4 factors. So, and they managed to alleviate a lot of hallmarks of aging. They did that on progeria mice, and also managed to increase their lifespan. So progeria mice live around 3-4 months only, unlike 2 years of normal mice. And they managed to extend that lifespan. And a lot of the, on cellular physiologic level, they checked many different features that looked like normal, and they didn't have also markers of senescence. And there was, importantly, no teratoma formation. So they did that through repeating 2 days of expression of the Yamanaka factors, 5 days of pause, and then they repeated that 3 to 8 times in their experiments. And this is the result they got, and it was, very exciting study, I think, for many at the time, and for us including. And we thought, okay, it's all very exciting, but because we don't know, like, whether these marks, transcription or some of the other cellular physiological features might be just temporarily improved to look like young. Is that actually a long-term change? They didn't follow up for a long time, these mice. And how do you know that they actually rejuvenated the mice? So the epigenetic clocks have already been out, but there was no clock for the mouse at that time. So they couldn't check if they actually measured if they actually rejuvenated the methylome. So we wanted to, thought what we can do to to answer these questions. Can we see if that can happen in a human cell? And is there a way that we measure it actually with the epigenetic clock? And then how does that, because they measured different markers, like they showed that the mouse don't develop cancer, but what happens with the cell identity in the end? We don't know. Like, do these cells actually start dedifferentiating in the mouse or not? And I'm just gonna open a bracket for the epigenetic clocks, although I think most of the people here know quite a lot about them. So the 2013 is the clock that was published by Steve Horvath. The multi-tissue age predictor is what really changed the things. There were some clocks based on 3 or like fewer CPGs a bit earlier, but that really changed everything because that was multi-tissue, worked very well, very, very robust, very accurate, just like 3.5 years of error. And the best telomere clock until then was around 10 years of error. That was a huge improvement. And it's based on, like I said, a simple machine learning algorithm. And as you see, it's only follows 353 CPGs. So just when people, or even our students, when they say, yeah, the metallome is very dynamic, you have 30 million CPGs, and the clock picked up 300. So a very, very small proportion would be the ones that change with age. The rest just doesn't change. Of course, this is based on the arrays. So the array itself, the one that Steve Horvath used, has 27K. So they're not the 30 million that exist in the genome, but still out of 27,000, 300 actually only correlated with the age. So it's a very small percentage, which is good because I think that's what's the beauty of the metallome, because it's quite stable. But small change do happen, and when they happen, they can also change quite stably and be a reliable marker. So they're based on the methylation the methylation values of a selection of CPGs that correlate with age, although we know that some of the CPGs in specifically Horvath's clock and other clocks, some of them don't change. They flatline, but somehow the algorithm picked them up because they maybe correlate with other CPGs and stabilize the clock. And the algorithm basically assigns different weights to the CPGs to then calculate how much weight or importance each CPG would have in the algorithm. The human clocks are usually array-based. I mean, the ones that we have, and Steve Horvath used the older arrays, which are 27 or 450K. Currently, the EPIC array is used, which is 850. And maybe there'll be 2 million or bigger arrays coming soon as well. But even with the large arrays, still the clocks usually pick up a handful, few hundred CPGs that correlate with age. The non-human usually based on other technologies, RBS, just reduce representation genome sequencing. Some clocks are minimized, like they only use a few CPGs to be easier. And some are composite. Apart from the age, they're trained on other physiological markers. And they're more predictors of mortality and morbidity rather than aging. But it is at the moment the best prediction. We don't really know what they measure, and actually the different clocks measure different things because sometimes on the same samples and patients they show different results. But what we know is specifically for the Horvath multi-tissue clock, it correlates with different conditions that are known to already have increased accelerated aging, like Down syndrome, HIV infection. They also are different for the tissues. For example, breast tissue is known to age quicker. Menopause increases aging in women. Men on average age a bit quicker than women, etc. Differences between ethnicity. So it does capture degrees of biological aging that we already know and have been known before that exist. So it's considered that it does measure biological aging, but we don't exactly know why and what it measures. So the study, to answer the questions I showed before, we decided to find, we wanted a quick answer, and we were in a reprogramming lab. So we decided to look for data which is publicly available and would have DNA methylation data based on array, so we can run the clock algorithms on that, but also would have gene expression data so that we can correlate and compare, what the gene expression is to assess the somatic identity or pluripotency, identity, how this dynamic looks. And so this data set was, we chose data set from Yamanaka and Takahashi's group again. So we thought that's a good one because they know their system very well. It's very well described system. So we could draw from different papers how these cells behave in the different time course which they've used throughout different papers. So we knew a lot about these cells, and they sorted them for a marker for pluripotency. So because the efficiency is so low in this process, in order to characterize the cells that reprogram better, they have to sort them through FACS, which was described in previous presentation. With fluorescence markers, they pull the fluorescent cells apart from the others, and then only the fluorescent cells continue growing, so that you have a selection of the ones which were successful in the process. So when we looked at the data, it basically turned out, like, we knew that pluripotent cells have 0 epigenetic age because Horvath already showed it with the paper that he published, the clock. But like, no one knew at the time how that happens. What is the dynamic? Maybe it's flat and then suddenly it drops. Maybe it's flat almost until they're already pluripotent and only then it drops. We didn't know how it happens, but we had hopes that if we see different dynamics from the dedifferentiation dynamics, that we could basically see that de-aging is different and decoupled from dedifferentiation. So then that can be tackled separately, and that's what we saw. That it started from more or less the beginning, quite quickly going down, and by day 20, the epigenetic age was reset to 0. While the full-time course is 50 days, so you don't get the cells fully pluripotent until day 50, but the age was already 0 at day 20, meaning actually that happened very quickly and before the dedifferentiation took place. And the change was dramatic, around 30 years, 35 before the point of no return. So we looked at this. The point of no return is kind of a state defined that until this point, cells need the Yamanaka factors. After that, they don't need them anymore. And if you remove them, they will continue towards pluripotency because they cannot return. The epigenetic changes or reprogramming has advanced so much that they're not turning back. So of course we wanted to get before that point, because before that point the cells, when left without the Yamanaka factors, they can spontaneously revert back to their original somatic state. So for us that meant that they haven't lost their identity. Even if it's temporarily suppressed, they still keep a memory about it and they can return to back what they were. So we were interested in that window, which is with the yellow, the so-called partially reprogrammed window, where we see that actually they're safe. That's still well before the point of no return, but we see substantial rejuvenation, drop in epigenetic age already happening there. With different clocks we tried, and they showed similar trajectories. They all reset at day 20. Although there are differences between how quickly that happened, and actually for most of the clocks it was quite slow in the beginning and faster towards the end. So for us, we thought that maybe in the first week not much happens, or first few days, and then the interesting bit is in the second week. Although recently there was another clock which was on deep neural networks based, so not the linear regression algorithms that were used before, and called AltaMage. And actually AltaMage run on the same data shows that the drop happens immediately and it's much faster. So there's probably an artifact, or we don't know, or just the AltaMage captures different a different aspect of aging from the Horvath clock. And it shows that it actually happens much faster, which is, I guess, better news for rejuvenation, because it does happen quite quickly. So then we looked at the genes, the gene expression, just to track and see how gain of pluripotency occurs. It has been described before that there are early and late pluripotency genes, and we saw exactly this, that a group of genes were going up faster, another group is going up slower. And so these are basically, the direct targets of the 4 Yamanaka factors, because they're transcription factors. So the genes that they immediately activate, and then the late genes are already downstream of this first wave, genes that are activated by the targets of Yamanaka factors. And we saw that they are not up until nearly day 30. Maybe kind of already day 20 they're quite high, almost same as pluripotent levels. But that was already where the clock was reset. So basically these genes were not at pluripotent levels while the rejuvenation was happening. And that was very exciting for us, because it meant they're actually they're stepwise rather than in parallel. The de-aging happens first, and then once the age is reset, already identity is removed, so to say. And then the fibroblast expression. So, in the previous slide, we looked at the pluripotency genes, how they get up. Now is the identity of the fibroblast, how quickly is that erased? And we actually were surprised to see, we expected the gene expression will go down, but we again tried and clustered the genes in big groups just to get the big picture, how they change. And we saw that one group remains fairly stable, actually up until day 15. And this group contains some very key fibroblast factors, collagen, which is something that fibroblasts secrete, and fibroblast-specific protein 1. So these were markers that were markers of fibroblast identity. So even if other fibroblast-specific genes drop, we see that until day 15, they kind of hold on to their identity and don't really let it go. So it might start being suppressed, but they hold on to it for longer. And which really surprised us, because you see that in the first week there's a drop, and actually after that there's a plateau, and they kind of stabilize. We knew that senescence has been described that reprogramming activates senescence because it's perceived as a stress signal to the cells to become suddenly embryonic. And could be that this plateauing of the fibroblast identity is retained because of senescence signals that kind of try and hold and stop the reprogramming. So possible, we don't know, but that could be an explanation why the fibroblast genes plateau and don't just continuously drop. So this was this is like a summary of the different findings we have. Maybe age rejuvenation in the orange. Maybe we can extend even to the beginning if we trust the new AltaMage clock. But basically, yeah, we were excited, and with this study wanted to get the idea out that reprogramming first rejuvenates and it differentiates afterwards. It's like a two-step process. And they are quite probably separable from each other in this way. And because this was one of the few, after Ocampo's papers, there was not that much happening. And then we got this paper out, and it's think it stirred a lot, commentaries and reviews, and other papers started coming out to show similar things. So I've kind of spoke about the things that led to here, and I'm just going to do a quick overview of the following papers. So that's a bit more detailed kind of timeline. People said and already started making timelines of what led to the partial reprogramming discoveries and who discovered what first. So yeah, I kind of said until 2018 when our paper came out. Then I'm going to go quickly through the papers afterwards. So right after our study, Sarkar et al. So this is a group from Stanford. They did a time course as well on two types of fibroblasts, endothelial cells and aged fibroblasts. And they did 4 days of Yamanaka factor expression, but it's again the longer set, the 6 genes rather than 4. And they did 4 days of expression, then waited for 2 days and analyzed the cells. However, they use a different reprogramming system, so theirs is supposed to be 2-3 times faster than the one that the team of Yamanaka used, the one that we analyzed. So this should, if we have to map it onto our trajectory, it should be probably more like first week rather than 4 days. However, so they see that the gene expression changed. So in the treated cells, it resembled the young fibroblasts rather than the aged. And they also see change in histone marks like H3K9 methylation, HP1 gamma, so a similar protein to the one that Prim Singh described with the very first proof of concept. They also checked the epigenetic age. However, what they found was that it was barely significant. And actually the Altum age, the neural network clock, doesn't detect any change in their data. So it could be that it's a transient effect, because if it didn't get down to DNA methylation level, it's probably not stable. It just didn't get rewritten. The methylome is not changed. So these changes likely will not persist. So there was another paper that came out last year as a preprint and got published recently. So they kind of followed the time course that we did with the same cell line and the same 4 Yamanaka factors, and it's the same length. So that's why I'm showing our plot from our review, just to lay it over kind of our described experiments as well. So they focused on only the middle, this boundary of the safe versus non-safe and point of no return. So they did days 10, 13, 15, and 17, and they kept them without Yamanaka factors for a month, and then analyzed them. So that was to see whether these changes that we observed in the reprogramming course of Yamanaka and Takahashi, whether these changes would persist after 1 month. And they've analyzed different aspects. They also show that they get very similar trajectory, around 30 years of epigenetic years rejuvenated in that span, in that point in the time course. And age was dropping, transcriptional age, youthful genes went up, and the mobility as well of the fibroblasts, they checked different things. I'm just going to show quickly a couple of things. That epigenetic age, which is on the first plot, that changed. So the purple are the rejuvenated cells. So they showed that it's down in almost all of them. Actually, day 17, the one that was longest with the time course, doesn't have a change. But the optimal seems day 13. And also with the transcription age, kind of like a clock based on the transcriptome, they also see that the first 2 time points, 10 and 13, worked better than the later time points. So they suggested that the sweet spot for rejuvenation is day 13, which actually coincides kind of with just before the point of no return in our time course as well. So kind of that they trying to go as far as they can and as far as things still work, to get the largest effect of rejuvenation. And they've analyzed transcriptome and global methylome and showed that, so again, the purple ones are the reprogrammed. They basically cluster with the controls, because the dark blue are the pluripotent cells, so they're very far from them. While the pink cells, the pink cells are the ones with the numbers of the different days. So these are the cells that were in the time course. So they were measured at the day they were kind of stopped from Yamanaka factors. So you could see that they've went a bit further towards pluripotency, but then came back a month, when the factors were stopped for a month and they were grown in culture, they reverted back completely to their original identity. But they remain rejuvenated. So a number of their genes are more similar to younger. For example, collagens, which, as I said, are very typical of the secretome of fibroblasts. The fibroblasts of the rejuvenated purple cells are more similar to younger cells than the aged. So then going on to another study. So this one is from a company, Calico Life Sciences, actually, and they've done the first single cell analysis of a time course. However, their time course is quite short. So they did two cell types, adipogenic and mesenchymal stem cells. I don't know why they took stem cells. I'm always, when they're progenitors, I'm always more kind of suspicious. Okay, well, they're already one step closer to pluripotent. I mean, they're multipotent. So they haven't taken the fully differentiated, terminally differentiated cells. And the idea, which I personally don't agree with, but I'm presenting to you, is that after the 3 cells, they lose their identity. And that's just the first 3 days out of a full 50-day time course. And then they claim that they redifferentiate de novo after they've lost their identity to become what they were. To me, what they lack, so this is still a preprint, they claim this on the basis that they don't compare to pluripotent state. So they of course, they do see changes in 3 days, but when you don't have the pluripotent state kind of there in your analysis, in your UMAP or where plots they used, then how do you know? If you have pluripotent cells, maybe all of these cells will be kind of one in one corner together and very, very far from the pluripotent state. So if you don't have your right control, they don't calibrate their plots right, their scale. So, but it's very good because it's a single cell and you can see the dynamic, because all these studies were done in bulk in the cell population together. And we know that some cells will reprogram much faster. So if you are to do that in a human, then what if you need 1 cell to get cancer that actually reprogrammed faster and became dedifferentiated? So single cell studies are quite important to see the dynamic in the population, in the individual cells. Like, do we have cells that actually reprogram much faster? And so where can we stop? How far can we go? Maybe 13 days would be too long. And what they did, and was nice, is they started screening for different factors. So they have the 4 Yamanaka factors, but all sorts of combinations between 3 of them or 2 of them. And they see that actually, even with 2 factors, they get very similar effects, and with 3 as well. So you don't need the 4. And they also tried with a completely different factor, MSH1, which is a multipotent. It has been published before that it helps myoblasts differentiate. So they just introduced this in myogenic cells, and that helped them regain back cell division properties. So they managed to rejuvenate them only with this factor, which was specific for this cell type though. But they show that, yeah, you can use factors which are different from the Yamanaka factors. They probably don't bear the risk for cancer, but they can achieve rejuvenation. And I think that's where a lot of labs work on this now, and is to find the rejuvenation factors. Just one slide for one of the in vivo studies, the first in vivo study after the Ocampo progeria study. So from the Sinclair lab, they used only 3 of the factors, and they managed to rejuvenate optic nerve of a mouse. So they damaged the nerve, and then they introduced the factors, damaged the nerve, and it rejuvenates when it has these factors versus not having them. And they showed that the methylome also changes. See if the injured cell is like the intact cells rather than the injured without the factors. And they also show in the last plot that depends on one of the enzymes of the methylation machinery. So the enzyme that can oxidate methylation in adult somatic cells, if it's not there, the rejuvenation doesn't happen. So the reprogramming the rejuvenation depends on demethylation. And I'm just going to summarize. There's a few more studies that show in vivo effects, but actually quite a lot of them focus on regeneration. So because regenerative properties are part of younger characteristics, a lot of groups, naturally, they focus on, can we regenerate this? And then we show that it's younger. It regained back younger properties. So introducing Yamanaka factors can reduce fibrosis and help the skin rejuvenate better, recover better without forming scar tissue. And actually 2 studies have looked into that. Then a few have managed to rejuvenate chondrocytes in humans with osteoarthritis. Muscle stem cell potency is reversed in skeletal muscle, but also in cardiac muscles. Cardiomyocytes have been turned back into more progenitor-like state where they can divide in order to recover the heart after myocardial infarction. But all of these focus on regeneration, which kind of though is like what Yamanaka reprogramming was meant to do anyway. That's part of the being back, returning them back to more progenitor-like state maybe. And they have not looked at rejuvenation per se, like in increasing lifespan. So a study that came out just recently actually, and that's a follow-up from the same study in 2016 that they did rejuvenation on progeria mice, but that's with wild type from the same group, wild type mice. So they don't see increase in lifespan. They did the same system with 2 days of Yamanaka factors, then 5 days of pause. They did that for much longer. So one of the experiments was 1 month, more similar to the original paper. Then they did for 7 months and 10 months. This cycle repeated, for 10 months in the mice. And still they didn't see an increase in lifespan. However, there was some effect on DNA methylation, including to the aging clocks, but it was mild to moderate, not very strong and not very convincing. So it doesn't seem to change the methylation and the aging signatures long term. They did manage to restore youthful gene expression, to reduce inflammation and SASP. SASP is the senescence secretome that you get from the senescent cells that is toxic. And they improved the regenerative capacity of the skin, which was also, and the fibrosis and scar formation, which was shown in another study as well. And they restored, in serum, all these metabolic and lipidomic profiles that distinguish younger from older blood. They managed to restore this in the mouse. So quite a few of the aging features were restored, apart from really DNA methylation and lifespan. which probably means it's not very long term. And then just the last paper that is still a preprint that also looks at extending lifespan, at rejuvenating specifically. So they repeated this with progeria model. Progeria is, I didn't mention, like a syndrome for accelerated aging, like they have senescence and accelerated aging. So they did that with constant expression of the Yamanaka factors or with a single shot. So to the left is the single shot where they did 2.5 weeks of expression of Yamanaka factors when the mice were 2 months old, and then they stopped. And they followed them till the end of their life. And that did increase in progeria mice, the lifespan, but also increased more modestly, but it increased lifespan in wild-type mice. So they managed to get them, normally they live till 2 years, and you see there's a quick drop around 2 years that a lot of mice die. Very few remained living longer, but they actually managed to get a much bigger proportion of the mice living until nearly 3 years. So if that happens for humans, that will be a big thing. That's big increase in average lifespan and health span. They did a lot of analysis, which I'm not going to go through, like all the organs, like literally in bones in these mice they checked to show that they look like younger. So as a summary, I think so far we can agree that it's more or less an established method for rejuvenation in vitro specifically and potentially in vivo with these last studies. That rejuvenation is achieved by a global remodeling and rewriting of the epigenome, which includes the machinery of putting and removing the DNA methylation marks. Short-term treatments show to be less efficient and maybe transient in comparison to the longer term, where the Yamanaka factors were just kept for longer before they were removed. So more than 10 days achieve already change in the methylome. That so far we see that rejuvenation can be applied to non-dividing cells, but it needs to go through cell division, restoring the cell division, which actually is logical because if we need to rewrite DNA methylation, you would need cell division and replication. And something I didn't talk about, but just to mention, there was a paper recently coming out studying the natural rejuvenation effect in mice, and very early on, just around implantation, they saw that is where the age is reset in every one of us. So unless the clock is affected because of these global changes of methylation that happened then, and unless this is an artifact, they kind of prove that the age resetting happens in all of us in early development before we start aging from zero again. So in essence, that's probably, actually encouraging because it means it's an experiment that happens in each of us all the time, and it actually proves that it's a long-term, has long-term effects. So just quickly on perspectives. So we still need to identify the boundaries which are safe, and they differ between the IPA systems used. And single cell studies will help us tell us more, what the dynamic is and if there are cells that are becoming dangerous and they differentiate quicker. The discovery of new factors which are non-Yamanaka factors and which will be safe to be expressed. We still don't know how stable it is, so the experiment that's been done now, it's with the human cells that they've been just kept for 1 month later, but potentially the preprint with the mice, they kept them for their lifespan and it was a long-term effect. But still more studies need to be done. And then for human interventions, we would maybe need non-genetic ways to affect the reprogramming. So with chemicals, maybe these SIMBA molecules, maybe something else, something that can maybe activate the genes or elicit the same effect in the cell without having to introduce genes and genetically modify the organism. Yeah, so this is the team that participated in the study that we did with the head of our lab and a student that was helping, and Ricardo Marioni is yes, a person that's involved in a lot of the clocks papers that were published together with Steve Horvath. And I thank you for your attention and happy to answer questions.
Yeah, we have some questions.
So I missed this at the beginning. How do you initiate these the Yamanaka factors?
So with vectors, just classical genetic engineering, you put vectors from viruses, retroviral or lentiviral systems, and then you introduce them. One of the papers actually used just mRNA based, like the COVID vaccine. They just get the mRNAs, and that was a much quicker system. Although, you know, that one, at least the DNA methylation effect wasn't strong. So it might not lead to long-term effects, or it may need, they only introduce them for 4 days, so maybe they need to keep them longer. But that's the way, you know, you put either genetic material or mRNA so that you start expressing these genes in the cell, overexpressing them for forced expression, because we have them in our cells, but, you know, they're suppressed, they're not expressed anymore.
So you said with like the Browdy study, the one you recently went over where they were doing the partial programming, and it shows, you know, it starts to have young characteristics, but it doesn't lead to extended lifespan. Did they, even when they did it out to like 10 months, and when they, I mean, I guess they observed them until they died, did they have like younger characteristics like for a long time, and just until their death, they were just like younger in general, and then they just die anyway at the same time?
So to be honest, I have to look specifically, because it didn't become clear to me whether they said they were till 25 months of age, you know, like 15 to 25. That was actually the plot where that's the schematic they used to keep them, like either from 10 months they introduced them and then take down age. So I guess they killed them at 22 or 26, 25. But they still show this plot for the lifespan, which could have been, like from their perspective, they showed it that it's safe, that they all lived as much as wild-type mice would live, and none of them formed tumors or died earlier. But it doesn't seem like they actually kept them to see if they live longer.
Okay, so it's not
Which
clear.
could be, yeah, could be an answer why they didn't see. I don't know why they didn't address it though.
I feel like
I'm
that was, like,
surprised.
important.
Yeah, so I mean, I have to check again because it's a big paper, but yeah, what they officially show is that there's no, if they had seen, they would have published, you assume. Like if they kept them for 25 to 26 months, why not keep them a few months longer to see if they will live longer?
Yeah, because if they were young, but they also, you know, just ended up dying at the same time, you know, that's important. You know, and maybe you need that. You said that it didn't affect their methylation age, right? Long term.
Yes.
So maybe that was more important than any young characteristics.
Yeah, so they actually don't see any effect with the Horvath clock and these clocks, the linear model based. So they used a different type of clocks that were developed more recently. The Look clocks, I actually don't know that much about them, but basically they didn't see much change with the classical clocks.
So with the partial programming, they removed like aging epigenetic markers, right?
Yeah.
So why wouldn't that be tracked on the Horvath clock? Doesn't that look at the same markers?
Yeah, well, I guess it just doesn't happen with this method. So the time course that we checked, it's exactly the same factors. I mean, first here is the mouse. So the mouse clock is not, the mouse clocks are maybe less robust than the human clocks, so to say. There are a few, they work quite differently. So it could be that that's why they use different clocks. But yeah, all the mouse clocks also pick up different CPGs, different combinations of CGs, different sets. My idea is that actually DNA methylation doesn't change much. And I think if you express just for 2 days and stop, I don't think it ever goes to global methylome or epigenome remodeling. So the changes would be, they see changes until they keep this cycling of the factors. So if they culled the mice when they stopped the Yamanaka factors, then yeah, they would see this change because they kept them. But I think if they keep them for some months afterwards and check then, it might just mean that things have gone back to where they were. That these changes are more short-term rather than long-term.
So how do you adapt to that? How are they going to make it more long-term?
So I think the other studies that do it for longer, that's what they show. If it's for around 2 weeks, either Jill et al. Study, they do it on the human cell line, like recapitulating our time course and choosing the time course around the 2-week point of no return mark. They have a longer than that other study in a preprint. They also have for 2.5 weeks expression, so that's a much longer one-off expression that would drive probably a deeper level changes, because I think they see stronger changes and definitely see change in lifespan as well. Not as, like in a wild type, is not as drastic as you see in the progeria model, but it is significant. And I think, yeah, that's the answer, just longer term interactions. Some are too short. I mean, who knows, for human or for therapies, maybe these short ones are safer, and maybe that's all you need to keep yourself a bit younger, but not risk anything. But we just don't see that going that deeply into the epigenome.
I would like to see somebody do Yamanaka factors in the hypothalamus, just combining two ideas. Is that feasible?
So it has been, I think in mice, they've done, but not the hypothalamus. It has been done in brain indentate gyrus cells. So I'm not sure, but that would be possible with these mice which have the expression, which are genetically modified to express the Yamanaka factors. But I have to check. I don't think anyone looked specifically at hypothalamus. That would be interesting because these cells are really very terminally differentiated. And yeah, if you can get them back to cell cycle, to resuming cell cycle, would be a lot. But then I don't know if that won't cause tumors to get your neurons replicating again in the brain.
We don't want them to cycle. We just want them to lower their inflammation level, according to the work of Dongchang Tsai. That inflammation in the hypothalamus is a driving factor for aging through the whole body.
Yeah, we just don't know whether that's because all of these studies show that you have to restore cell division in order to do that, to rejuvenate. We don't know whether it's possible. But then in the brain, I think inflammation probably doesn't come from the neurons, but from the glial cells. So then that's different. Maybe they can be manipulated much more easily. I think that paper on the brain, which I didn't include here because they're getting more and more, and it'll be too long to include every single paper. I think they looked at glial cells. Again.
I'm interested when you say, I always thought men die or you only live 8 years less than women. I always thought it's because they get more premature atherosclerotic heart disease and other things. You say, it's interesting you mentioned that they age faster. Are you just assuming because they get these age-related diseases, or there's evidence they have more methylation, in their DNA and things like that?
I don't know about methylation levels per se, but these were just measurements in cohorts, and they probably saw that in men generally, their age accelerates a bit faster.
By the Horvath clock.
But with the
they age
Horvath,
faster than women.
yeah. So I don't think that was related to car crashes and these or diseases. Yeah, it's just measurement based on the epigenetic clock, which is usually from saliva or blood samples.
So they do alter epigenetics. Is that what you said?
No, just maybe their clock is ticking a bit quicker.
Oh, okay.
But that will be a small difference. It's not a very big difference.
I was kind of curious when you just mentioned something about transient. I think it was with the viral vectors thing that we're speaking about. So when you introduce the Yamanaka factors, are those going through viral vectors, and are they integrating the Yamanaka factors into the genome, or are they just like transiently expressing or overexpressing the genes? And I guess how do they manage to only keep cells overexpressing the Yamanaka factors for only a certain amount of days?
I think the retroviral factors, vectors are integrating, which is part of the thing that these papers discuss, that it needs to be a system that doesn't integrate, which the RNA system that one of the studies uses. That's what the benefit that it has, that it doesn't integrate.
Your epigenetic age increases. It's an increase in the age-associated epigenetic marks, or just different marks?
It's only the ones which are in the clock. Like, we use the clocks, so it's only the CPGs which are the ones that the clock algorithms picked up.
And the more methylation corresponds to more age, or is it just different methylation per?
It's different because some CPGs go up, some go down in the clock, and it kind of calculates. So it's both ways, and some don't even change in the clock. They just stabilize it.
And the point of no return, do we know what happens there?
I think so. That's the idea is that the cells have already gotten to sufficient remodeling and change that they can't turn back. So this is usually related to the SOX2, the endogenous SOX2 expression and downstream genes. That when they kick off, the process is basically reversible, because it starts like a cascade of genes that activate other genes and other genes. So that's already the effect that's a few steps down from the first genes that the Yamanaka factors activate. That's why it takes some time and kicks off afterwards. And of course, not in all cells. It's in the ones that really continue reprogramming.
Okay, let's thank the speaker again. Thank you so much. The characteristics of an organism are adaptations to its environment. This was Darwin's foundational observation. He observed that the beaks of the various finches he encountered were well suited for the food available in their environments, because the characteristics of the individual organism are adaptations to its environment. It should thus come as no surprise that aging is also an adaptation to the environment. And there's no reason we have to age. There's no reason we can't get stronger and healthier with the passage of time. But aging is built into our bodies. And partial reprogramming is just one method, but sufficient in itself to convince you that we can intervene in the chemical processes that are making us age and cure aging in our lifetimes.